Hi ladies, this is the end of chapter 14 on mutations. So what exactly is a mutation? Well, a mutation is just any change in the genetic material. The first thing we're going to look at is a point mutation. So this is a change in just one nucleotide pair of a gene, and sometimes this can lead to an abnormal protein. For example, in the case of sickle cell anemia, there is a point mutation right here in the mutant hemoglobin DNA. This one change right here from T is going to cause a change. Okay, so this one mutation here is going to cause a change in the mRNA, which is then going to cause a different amino acid to appear in the protein sequence. So you can see with normal hemoglobin, there should be a glutamine. In sickle cell anemia, the individual has a valine in an extra valine in their hemoglobin molecule. And because there's an extra valine, the protein isn't going to fold correctly. You can see here's normal hemoglobin with the 4 polypeptides working together. And you can see here with sickle cell hemoglobin, there is a complete change and there's almost like a linearization of this protein complex, which is going to change the shape of a red blood cell from a nice circle that can carry lots of oxygen to a sickle-shaped cell that cannot carry oxygen very well and sticks together, which can clog little capillaries. Okay. So that was a case when a mutation could be bad. Some mutations are not bad, however. Mutations can be good. It really just depends on the phenotype that is created. If that phenotype is beneficial, then that mutation is a good mutation and will be maintained in the population. If that mutation is bad, like with sickle cell anemia, usually these mutations are not maintained. In this case, however, there is that heterozygous advantage that we talked about in the genetics section of this unit, so this trait has been maintained in the population. Mutations can also be not good or bad. Sometimes they're just silent and they don't change anything. I'm going to show you an example of silent mutations, missense mutations, nonsense mutations, and I'm going to show you what insertions and deletions can do as they can cause frame shift mutations. So here's the silent mutation. You can see here, here's our original wild type. You can see here there's an A instead of a G, which is going to cause a U instead of a C in the mRNA, which is going to not do anything to our protein. So this is what we call silent. Okay, glycine is still in this amino acid right here. Okay, that's because the codons are a little bit redundant at times. Sometimes silent mutation can occur. With missense, you have a T instead of a C in this position, which will cause an A instead of a G in the mRNA, which will cause serine to the amino acid serine to appear in the protein. Okay, this is a missense, still coding for a protein, but you're going to have one amino acid that's a little bit off from the wild type. Could be good, could be bad, depending on what this protein actually is. Here we have a nonsense mutation, A for T, so in the mRNA you get U instead of A. This is going to code for an early stop codon, so this is a nonsense mutation because it basically forms a truncated protein, or maybe even not a protein at all, depending on where that stop codon is. So this is going to form a non-functional protein. Over here we have frame shifts. So in this example up top, we have an extra nucleotide. So this is an addition or an insertion. And in this case, the insertion caused an early stop mutation, or an early stop codon, excuse me. And so this is a frame shift causing an immediate nonsense. In this example, we have a deletion. So this one is going to this protein is going to continue. It's not going to have the stop codon in the appropriate location. So this is going to cause an extensive missense, okay, make a protein that's a lot longer than it really actually should be. Just adding one nucleotide can do that. In this example right here, there are three nucleotides are missing from the protein. And what this is going to do is going to remove an entire codon from the mRNA. And because it does that, it's going to remove an entire amino acid from the protein. So you can see here, it goes methionine, phenylalanine, and glycine. We're missing lysine from the original protein. So sometimes removing more than one or adding more than one can change the protein substantially. So a lot of DNA mutations are spontaneous, just with replication, recombination, repair. It's very easy for a little mutation to slip in. Usually these are caught and corrected, but occasionally this mutation can appear in the DNA. There are chemicals as well that can cause mutations. We call these mutagens. Most carcinogens, so anything listed as a carcinogen is which would cause cancer, is also mutagenic. And just to kind of bring this back to the idea of gene expression. So say we have a pathway, okay, that produces some product. If you have one mutation in one gene of that pathway, you're not going to have a functional enzyme, and then as a result, you're not going to be able to produce the product. So mutations can not only affect the expression of one protein, but they can interfere with an entire gene pathway. Okay, so this was mutations. Please bring any questions you have to class.